Hello friends, this video on food production enhancement part 10 is brought to you by examfear.com. No more fear from exam. So let us now look at the factors which define improved plant types. So what are the characters which tell us that this is a good plant type or this is an improved plant type? So what do we want? What are the desirable characteristics which we look for in any plant type? First of all, increased crop yield. So we want more and more crops to be produced. So the productivity should be very high. Disease resistant plants. Now, disease resistance, when we say that the plant should have resistance against the pathogens, that is the disease causing microorganisms, it should also have high tolerance or resistance for the insect pests so that they cannot spoil them, because a lot of plants get destroyed by the pathogens or diseases, and that is why it is very important that improved plant type should be disease resistant. Increased tolerance to adverse environmental conditions. So what do we mean by adverse environmental conditions? So when we say that, we mean extreme temperatures. Maybe if the temperature is very high or the temperature is very low, the plants sometimes they are not able to survive. But if the plants have increased tolerance to changes in temperature, then it is definitely an improved variety. Again, it should be able to have increased tolerance for a situation of drought, that is when there is no water, or with salinity, if the salt content in the water increases too much or it decreases too much. So in those extreme conditions, the plant should be able to have high tolerance. That means it should be able to tolerate up to a certain value. So now we will look at the steps for plant breeding, how exactly plant breeding is done. So that is what we will study here. Now the following steps are carried out for breeding plants. The first is collection of variability. So variations, why do we need variations or varieties? Because when we want to look for the desirable characteristics, so in order to look for desirable characteristics, you need to have a collection of all the possible varieties of that plant so that you can choose from that variety whichever is the desired trait and whichever trait you do not want. For example, you want to go shopping. So when you go for shopping, let us suppose you want to buy a dress for yourself. Now, if the shop doesn't have a variety of dresses, then will you buy it from there? Let us suppose the shopkeeper says that he just has one dress. So will you just look at that one dress and purchase it? No. Right? Because you would like to see varieties. Maybe you will go to some other shop which has some 20 to 30 dresses of different types, so that you can look at all of them and then decide which one you want. So in a very similar way, you should collect all the varieties of plants so that the desired trait can be chosen from them. Now this is the first step for crossing a particular crop. That is, this is like a prerequisite. Now this collection of plants for a particular crop is known as germplasm collection. So this collection is termed as germplasm collection. So this collection will actually have all the alleles for all the genes of a particular crop. So now the next step would be evaluation and selection of parents. Now from that germplasm collection, you actually need to select the desired traits. For example, when you go to buy a dress, if the shop has some 20 dresses, you just need to choose one out of them. So in a very similar way, here also you have to look at the entire collection and then you have to select the parents. Parents means the plants which you want to breed. So you have to choose the male and the female plant which needs to be breeded or mated together. So desired characters need to be selected. So if you take the example of sugarcane, the sugarcane example which we took some time back, the one which was, so let us see. Let us suppose this is the first variety. This is variety number 1, this is variety number 2. So let us suppose that the sugarcane of variety number 1 has thin stem, whereas the sugarcane of variety number 2 has thick stem. Again, variety number 1, it has poor sugar content, and variety number 2 has good sugar content. Again, variety number 1 can grow in north, whereas variety number 2 can grow in south. So now these are the various characteristics of both variety number 1 and 2. So now you have to select the desired traits out of them. So in a very similar way here also, when you have collection of all the varieties, you actually need to select the traits which you want. For example, if you want it to grow in north, you want the stem to be thick and you want to have a good sugar content, so you will actually need to select those traits. So based on the traits which you want, you have to select that variety. So here we just have 2 varieties. Let us assume if we have a third variety which had thin stem, poor sugar content, and which grows in south. In that case, we will not select that variety 3. We will select variety 1 and variety 2 and then cross them with each other. So basically we are selecting the parents in this step. Now once the parents are selected, then what happens? Cross hybridization among selected plants. Now whichever parents we have selected, so breeding takes place. That is, crossing will take place between those 2 parents. Now how crossing takes place? The pollen grain of one plant is placed on the stigma of the flowers of the female plant. So that is how pollination, and which in turn is followed by fertilization, takes place in case of plants. So one's pollen grain should come in contact with the stigma of the other one. Now once hybridization has taken place, what will happen? Output will be produced. That means the offsprings will be produced. Now again, the offsprings or the outputs will have different traits combination, like as you have seen in genetics, like whenever a male and female are crossed, a lot of different types of offsprings are produced. So here also different types of new plants will be formed, but now we have to select the superior recombinant. So whichever output is obtained, out of that we have to select the desirable output. And then testing and release of new cultivars. So now what happens? Let us suppose the various outputs which we get when we cross variety 1 with variety 2. So let us suppose the output 1 that we get has thick stem, poor sugar content, and it grows in south. So that is output 1. Now let us suppose the output 2 which we get, it has thin stem, good sugar content, and it is good to grow in north. The third output, now this doesn't mean that these are the 3 outputs which are formed. I'm just trying to give you an example. Let us suppose that in any output, let us suppose you have these 3 varieties of outputs which are formed. So let us suppose the third variety of output which is formed is thick, good sugar content, and grows in south. So now looking at these 3 varieties, we have to select the superior variety. And which is the superior variety? Output 2. So this will be selected, and now testing will be done on output 2. So this output 2 will be tested for its quality. It will be tested to check if it is disease resistant or not. So it will be tested for a few growing seasons. Now, if it is found to be suitable, then it will be commercialized. So this phase is known as testing and release. So here it will be tested that, okay, if this is the final new variety which is obtained, so let this be tested. Let's see if it is disease resistant or not, or if it is able to survive well under that specific climatic conditions or not. So based on that, finally the new cultivars are released or commercialized. So this is how plant breeding takes place. Thank you. Please visit examfear.com for an easy 4-step learning process absolutely free of cost. Watch video lessons, ask questions, refer notes, and take an online test. Thank you once again.